Hey, well, welcome back to the Classics of Immunology Journal Club. Today is a very, very special day because we have guests, because we're going to review a paper from Rafi Ahmed's lab. We have also Rafi here, as well as his postdoc, Masao Hashimoto. Before we get started, I just want to remind you to like the video, click on the bell to get notified for more, subscribe, and then also check out my website, kendallasmith.com, because you can, on the writing tab, you can find all the classic papers there in immunology. So when late at night at 3 a.m. and you can't sleep because your experiments aren't working, you can read about experiments that have worked. So I want to introduce to you Rafi Ahmed and Masao Hashimoto first. Masao is the first author on this paper, and then there's 28 other authors, and then lastly, myself, and then Rafi Ahmed. And the title of this paper is PD-1 Combination Therapy with Interleukin-2 Modifies CD8 Positive T-Cell Exhaustion Program. This paper came out just a week or two ago in Nature, September the 28th, 2022. And last week, I reviewed a paper from 2022 and announced that for the first time since we started this Classics Journal Club, we now are in real time. So this is going to be fun going forward. And I'm going to try to do more of these kinds of situations if I can convince friends and enemies, frenemies and friends to come on with me so that we can try to explain these different papers. Masao Hashimoto is from Kumamoto University, where I've visited a couple of times, actually, back in the day. He is now a postdoctoral fellow, an MD PhD postdoctoral fellow with Rafi. Rafi has an interesting background. He grew up in India and went to college and in India, and then went on to a master's degree, and then came to the United States, to the University of Idaho. Must have been really culture shock. Was that culture shock, Rafi?
It indeed was quite a culture shock, and it was Idaho State University.
Idaho State University, yeah. Well, then he went on and he went to McGill University and enrolled in a PhD program after he got a master's at Idaho State. And then he dropped out of that after a while and then worked as in somebody's lab at McGill. And then was accepted into Harvard for graduate school, ultimately was there for several years, got his PhD, and then did a postdoc at the Scripps Clinic and Research Foundation in California. And I think he became under the influence with Michael Oldstone at that stage out there. Is that correct, Rafi?
Yes, my postdoc was with Mike.
He was your mentor.
From 81 to 84.
So that's where Rafi was introduced to the lymphocytic choriomeningitis virus, because Mike had spent a lot of time in developing this as a model system in order to be able to study the immune system, the immune response. That turns out that was critical because Rafi was smart enough to know and to see the future. And he has spent the last decades developing this model system in order to study exactly how the immune system deals with the microbe, in this case, a virus. And we, as the immunological community, have been the recipients of all the knowledge that Rafi's lab and his trainees have generated over the years. So the background on this paper is, the background really goes back to the 70s when people first started reporting that there was something wrong during a chronic viral infection as opposed to an acute viral infection that usually the experimental animal could get over. And back in the 70s, for example, and between the 70s and the late 90s, the field really couldn't progress. And it wasn't until John Altman developed the tetramer technique in order to identify and enumerate and so forth antigen-reactive, antigen-specific, peptide-specific T cells. The real first paper that put this field on of what's going on in chronic viral infections started in 1998 from Rafi's lab, and Zajac was the first author on a paper that entitled Exhausted T Cells During Chronic LCMV Infection, Persistence of Cells Without Function. And that became known then subsequently, or even before that, as the cells became exhausted, because they couldn't tell whether the cells had just disappeared, they died, that had been antigen activated during an acute infection, and so that the animal was left with no defenses, or whether or not the cells were still there, but they were not functional. This paper established the fact that they were functional. Then 5 years went by, and it wasn't until 2003, and I became involved with Rafi's lab at that point in time. And I remember I heard him give a talk at a meeting, and I said to myself, this guy's really smart. And so I invited him to come to New York City, where I was at Cornell, and we went out to dinner. I took him out to dinner. I always like to go out to dinner with visiting firemen, and proposed to him that maybe interleukin-2 was very important in this whole chronic infection story, and also in the acute infection. And so we went on to a series of experiments that was done by Joe Blattman, who was a student or a postdoc, I can't remember, in the lab at the time. And he did a really great job showing.
Joe was a graduate student.
He was a graduate. That's what I thought. Yeah. And he did a series of experiments. My contribution in this series of experiments was that I supplied purified interleukin-2 for the experiments, and Joe used the interleukin-2 to treat mice that had been infected with LCMV, both in the acute phase and in the chronic phase. And in the chronic phase, what he would do was he treated the mice for 7 days during the chronic infection, and he found that there was an increase in the antigen-specific T cells, CD8 positive T cells, and that was concomitant with a decrease in the virus. Well, I thought that was very important, and what I predicted, actually. And so that was probably my most intellectual contribution to all of these experiments. Then 10 years went by. Other things happened in the interim, obviously, but the next thing that happened was Erin West, as a grad student with Rafi, showed that if you combined therapy with anti-PD-1, that would synergize with interleukin-2 to increase the numbers of antigen-specific CD8 positive T cells and decrease the viral load. And that was published in the Journal of Clinical Investigation. Then 3 more years went by. 2016, Masao Hashimoto entered the scene. Actually, he was even there before that. And a paper out in Nature which defined CD8 positive T cells that respond to anti-PD-1 therapy and provided a proliferative boost because of the anti-PD-1 therapy, had the marker TCF1, and of course they were PD-1 positive. And that combination of markers enabled Masao and Rafi to segregate, to identify and then segregate the cells that were responding to their therapy. Then in 2019, in Immunity, there was another paper out that said effector-like transcriptional signature emerged from PD-1 positive stem-like, meaning they were TCF1 positive, CD8 positive T cells during chronic infection. So that's the background of this paper that we're going to look at today. And in the introduction to this paper, they make several points. The first one is that the PD-1 positivity of the cells activates a negative feedback pathway that plays a central role in promoting T cell exhaustion in chronic viral infections. So, and that's one of the reasons that anti-PD-1 therapy came to be used in the last decade in human cancer. And a lot of patients have benefited from anti-PD-1 therapy, and because they've activated the immune system to respond to the cancer cells. But they point out in the introduction of this paper that not all patients respond, depending on what kind of cancer they had. If they respond, they don't necessarily respond totally all the time, and they can also relapse. So the hunt has been on to improve this immunotherapy of cancer. It's been known that the PD-1s expressed on the antigen-activated T cells, and when it's activated, when it is there, it imparts a negative signal on the T cells that really essentially, at a transcriptional level, turns off cytokine gene expression. Interleukin-2 being one of the major ones, but as well other ones as well. So it sort of disarms the CD8 positive T cell. The stage is set for saying, well, I wonder whether or not if we combine interleukin-2 therapy and anti-PD-1 therapy, whether we can improve things. And so essentially there's a song out of the 50s, a popular song sung by Bing Crosby and the Andrews sisters. It's called Accentuate the Positive and Eliminate the Negative. And that's what combination therapy does with IL-2 and anti-PD-1. So that's the introduction, as we'll see in this particular paper. Now, the methods and the results I'm not going to go into in detail, only to say they used the LCMV model of chronic infection, a clone 13 of the Armstrong strain, and they examined using a model system that was worked up in Rafi's lab over the decades. They've treated these mice usually for 13 days, a couple of weeks, with anti-PD-1 to block the negative signal, interleukin-2, and PD-1 was given as an antibody subcutaneously or intraperitoneally?
Interperitoneal.
And then interleukin-2, 15,000 units, international units, intraperitoneally, twice daily for 13 days, or a combo of the anti-PD-1 antibody every 3 days and the daily administration of interleukin-2. And then on the 14th day, the mice were sacrificed and the analysis was performed. The combination had already been shown, I think Erin West had shown this, that the combination therapy was synergistic and it increased functional CD8 cells. And as we'll see, that happened in these experiments as well. And that increase in the number of, in the population of antigen-specific T cells, CD8 positive T cells, correlated with a decrease in the viral load of these chronically infected mice. That's very important. So one question that I started with was, can we show that the responder cells to this combination therapy are the so-called stem-like cells of TCF1 positive and PD1 positive? And so in order to do that, they would chronically infect mice and harvest the T cells from these chronically infected mice, sort them from exhausted stem-like responder cells, and they transferred those to clone 13 infected mice and then subjected those mice, because they could have allotypes to make sure that they knew which cells were which, either host cells or transferred cells. And then they treated them with the 4 different regimens, either no treatment, anti-PD-1, IL-2, or the combination. Let's go to screen sharing. And this is figure 1. And basically, it is the answers to the questions that I just posed. This is the PD-1, TCF1 positive stem-like CD8 cells provide the proliferative burst after PD-1 blockade, IL-2 therapy, and anti-PD-1 IL-2 combination therapy during the chronic infection. So that the experimental setup is in the A portion of this figure. So of the chronically infected mice, they harvest the stem-like cells and the exhausted cells, and then transfer them, and then treat them for 13 days, and then analyze on the 14th day. And you can see just from these plots of the flow cytometry that anti-PD-1 causes an increase in cells of the stem-like, and then IL-2 therapy does as well, and the combination does even better than either one of these two alone. In contrast, the exhausted cells are not responding to the therapy. And here we have the three different therapies, PD-1 therapy, IL-2 therapy, and the combination. And this is the donor cells per spleen. So you can see the untreated and then the treated. This is with anti-PD-1 therapy, and the exhausted cells, phenotypically exhausted cells, are not responding, whereas the IL-2 therapy is even a little better. If this is a logarithmic scale over here, and then the combination therapy is even better than either of these two therapies alone. So let's just move on to figure 2. So one of the questions that they really wanted to find out about was whether or not these stem-like cells responded to the combination therapy and became more like effector cells from an acute viral infection. And this figure 2, essentially says, yes, that's exactly what's going on. And it says that there's a distinct transcriptional signature of virus-specific CD8 positive cells after PD-1 combination therapy compared to PD-1 monotherapy during chronic infection. And so in the A portion, there is a plot where we're looking at untreated mice and then anti-PD-1 treated mice as compared to IL-2 treated mice in blue, or the combination therapy in red. These are different genes that have been studied, in the inhibitory receptors, effector genes, cytokines and receptors, and so forth. And basically, in this figure is summarized over here in a heat map kind of a situation. The effector-like gene signatures, anti-PD-1 and IL-2 combo therapy, or IL-2 itself, I was sort of proud of that, have got the effector genes on, and they also have memory genes on, whereas the exhausted cells are exhausted. And if you move on then to the D portion of this figure, you see that there's a cluster, what they call cluster 3. There's cluster 1, cluster 2, and cluster 3, and then naive in terms of antigen-specific GP33 peptide sorted single cells. This cluster 3 is what they homed in on, because in terms of the colors, you can see here that proportion of number 3 clusters in antigen-specific CD8 positive T cells, the IL-2 therapy and the combination therapy has got as much as 80% of these cells are in cluster 3. What they ended up concluding was that this cluster 3 actually defines the new LCMV specific CD8 positive T cells derived from the stem-like 8 positive cells after treatment with IL-2 and or the combination therapy. And they went on to look at, in figure 3, the functionality of these cells, the phenotype and the function. And the title of this figure is the phenotypic and functional characterization of LCMV-specific CD8 positive T cells generated by anti-PD-1 and IL-2 monotherapy and the combination therapy during chronic infection. And if you look at the subfigures A all the way through E, the answer is the cluster 3, which is now color-coded in the chartreuse here. The proportion of the 3 clusters in the antigen-specific T cells, you can see the cluster 3 is basically in the IL-2 treated group and the combination treated group. The phenotypically, there are different genes and gene products that are detectable on these cells in the chartreuse there. And then if you look at their functionality in terms of being able to produce cytokines, interferon gamma, TNF positive. Here again, we've got the function in the green. Here's interferon gamma and IL-2. And then CD107, they degranulate better in the combo therapy and then in any of these other therapies. So we've taken these stem-like cells from these mice and treated them with their magic potions, and they become very similar to acutely activated effector cells from an acute infection. Then they went on to look at the epigenetic signatures of LCM specific CD8 positive T cells generated by IL-2 or combination therapy. And they found that the IL-2 therapy and the combination therapies gave them different epigenetic signatures as compared to just anti-PD-1 therapy alone. And I think you can see that most easily here if you look at untreated versus anti-PD-1 and then look at untreated versus IL-2. You've got a whole cadre of genes here that are expressed, and both up and down expression. And then here is untreated versus combination therapy, and here's anti-PD-1 treated versus IL-2 plus the combination therapy. And with the combination therapy is superior. And then if you look over here at cytokines and chemokine receptors, you can see that the combination therapy, you have a lot of these genes on, whereas in the anti-PD-1 therapy alone, it's hardly there. They go on to look at also CD4 positive T cells, and they found that their therapy also improved the CD4 positive cells. And so, and that was CD4, it had been shown earlier papers that I didn't tell you about in the background of this whole phenomenon of the chronic infection. It got worse when there weren't any CD4 cells involved. And of course, CD4 cells, we found early on in our experiments with IL-2 that CD8 positive T cells and CD4 positive T cells, CD4 cells made about 10-fold more interleukin-2 than CD8 positive cells. So that didn't surprise me at all. And these new data sort of would fit right into that. The other thing that they looked at was whether or not the alpha chain of the IL-2 receptor that appeared on these combination therapy cells was important for the whole response. And the answer to that was yes. And of course, that's also called CD25. And I've never really liked that term, the CD terminology. It's great for molecules that you don't know what they do, but if you know what they do, you ought to call them the IL-2 alpha receptor chain. Anyway, the question that they wanted to look at, and this is in figure 5, is whether or not the alpha chain is important for the synergy seen in this combination therapy. And because there's a lot of work going on in the clinic, they've worried about activating first regulatory T cells. Of course, CD25 is markedly upregulated on CD25 cells, or IL-2 receptor alpha chain positive cells. If it's alpha chain positive, it's always, it's also beta chain and gamma chain positive. So alpha chain positive cells marks the heterotrimeric, very high affinity receptor. And when you have that high affinity receptor on cells, our studies have shown that the high affinity receptor is 100-fold higher than if you only bind the beta and the gamma chain. So with a higher affinity, low concentrations of IL-2 will work just fine, because they'll activate the trimeric receptor. So because these clinical trials have been trying to avoid that, so they don't get regulatory T cells, and they also were hoping that they wouldn't get many of the so-called toxic side effects that Steve Rosenberg had activated by giving very high doses of IL-2. And so what they found was that both cell populations, both the stem-like sorted stem-like cells and exhausted T cells transferred into congeneric mice so that they could track the transferred cells. And so here we have, this is chronic infection on day 0. Oh, no, this is just chronically infected mice. I'm thinking, so we got untreated, we have anti-PD-1 in the green this time, and the combination therapy in red, and the then treating with anti-CD25 to block the IL-2 receptor alpha chain. And you can see basically then they've studied the number of cells in the spleen and the liver and the lungs, and then the ability to make interferon gamma and TNF or interferon gamma and IL-2. And you can see in the brown here, when you add in an anti-CD25 antibody to block the alpha chain, essentially all the good stuff is decreased in these experiments. And so then that was what I expected anyway. But then if you go on to figure 6, they actually used one of the IL-2 receptor 2 mutants that doesn't bind to the alpha chain, but does bind to beta and the gamma. And essentially, you can see the same thing that I just showed you when you use the antibody to block alpha chain binding by the therapy. So I'm going to stop there. There are 13 extended data figures in this paper, each one of which has different subfigures. And so what I would implore all of you people to do, if you're really interested in this business, is to look at this paper carefully, see whether or not I've given you at least an introduction to this paper. And we'll stop there. Let's stop the screen sharing and go on to the discussion. They make several points in this paper in the discussion. One is, obviously, the combination therapy is more effective than just the antibody alone, which is what a lot of the original clinical studies have been using. The responding cells are the CD8 positive cells, are the distinct subset that are TCF positive, and they also are PD-1 positive. And those are the ones that they had shown earlier that give a proliferative burst when you give anti-PD-1 therapy. The combination therapy changes the differentiated program from stem cell to effector cell-like, and it's primarily due to interleukin-2. But it's also critical to block the PD-1 inhibition, and it goes back to Bing Crosby's song of accentuate the positive and eliminate the negative. The other thing that they concluded was the alpha chain expression and binding is important and essential has an essential role for the combination, the success of the combination therapy. And what I'm hoping, and I'm sure Rafi also hopes, is that the clinical people will pay attention to this particular paper and design their clinical experiments with the thought in mind that it might be a good idea to add IL-2 to the anti-PD-1 therapy. So I'm going to stop talking there. And sort of like, I don't know if you watch Rachel Maddow on MSNBC at night, but when she does one of these interviews to somebody, she'll go on and on and on and on. She talks a lot, just like me. And then she asks them, well, now have I summarized this properly to these guys? And so that's my first question to both Rafi and to Masao. If I left anything out, please correct things now before we get going any further.
No, Kendall, I think that's it's a very fair and clear summary. Summary is excellent.
Masao, what do you think?
Yes, that's great. I think basically 3 important points emphasized by your, explanation. Yeah, that's good. Yeah.
Okay.
Yeah.
Good. So, Masao, please add whatever you would like to do, particularly with the experimental aspects of all these studies. I was shocked about how much work there was involved in this. And I figured Rafi must have you in a cage. Rafi must have you in a cage out back where he doesn't let you go home.
So, I joined the Ahmed lab 10 years back or so. And before that, I have no basically experience for chronic viral infection model, right? Mouse model. I realized that it took so long to finish the experiment. Basically, we cannot make a mistake from the infection and wait, establish a chronic infection, then start treatment, then sacrifice, then analyze, right? So very long. So
and many chances for something to go wrong.
Yeah, so that is the most difficult part for doing experiments using chronic LCM model, because takes so long, right? Not like infections and quickly analyze because it takes maybe 3 months or so to know the answer. Then we can, reschedule or design the new experiment. Right? So that's why it takes time, and also physically a little bit difficult because tetramer positive cells are very low frequency in chronically infected mice.
Yeah.
So that's why it's a little bit difficult compared to in vitro experiments or acute infection model. Yeah.
Well, what is the proportion of these stem-like cells?
Stem-like cell, tetramer positive cells in the spleen is 0.1% or something. In spleen.
Really?
Yeah. Then among that, maybe stem-like cells are maybe 20 to 40% or something. So very minor for proportion. So for adaptive transfer experiment, sometimes we need 50 mice or something for getting donor cells. Yeah, that is, that is kind of one challenge for doing experiments in chronically infected mice. But that is, physical problem.
Right.
But another problem is we got a lot of data, right? And each data is very, if we describe detail, everything is different and new. But for writing the paper, we need to have a simple, clear message to deliver to the field, right? That takes, also a lot of efforts. To make simple but important message to the field.
Because
otherwise people cannot understand.
Because you have to do multiple repetitive experiments of the same kind of an experiment in order to get enough data to get the means or averages of exactly what happened, right?
Yeah, and also, we have always at least 4 groups or something, right? Or 6 groups of mice. Then we need a lot of mice to get a statistical significance. Yeah.
Well, I've always been impressed by Rafi as long as I've known him. He's a very patient.
That is correct. Yeah. For Rafi, maybe 10 years is maybe 1 year for general people, I think.
Yeah. No, that's exactly correct. Well, you must be also very patient.
I am fine. But just takes time. But I know that this study is important, and also because otherwise, if we didn't use a good model, answer is kind of misleading. Right? Data can be misleading if we don't use a correct model.
Yeah.
Yeah. So we need to use good model. And LCMB, chronic LCMB model is one of the best model to study chronic infection. So no way. I think. Yeah.
Rafi, you have anything to add?
Yeah, I think I would restate what Masao said. Masao put in an amazing effort in this project. He described to you that they're not easy experiments. Large number of mice, and in particular those adoptive transfer experiments, which were key in identifying which cell is responding and the conditions of this chronic infection after the PD-1 blockade and IL-2 requires really a huge amount of work. A large number of donor mice, the quality of the experiment has to be top-notch, and Masao has an amazing green thumb. Things work beautifully. But also something that Masao didn't emphasize as much, or he's fully aware of it, is that the analysis that was done was very detailed. We did not just the RNA-seq analysis, but also single-cell RNA-seq, ATAC-seq, all the functional studies that were done. So really it was a tour de force by Masao in terms of the functional characterization, in terms of the epigenetic analysis, and in terms of the transcriptomic analysis, plus all the biological experiments. So really, I'm very proud of this paper. It's probably one of the best papers we've published in terms of this comprehensive analysis. And as you pointed out in your very nice summary, Kendall, I think it makes several important points, and I'm kind of repeating what you've said and what Masao has said, is that the first important point is that the stem-like cells are not fate locked into a exhaustion program. And by providing the IL-2 signals, we can actually change that exhaustion. And I think to me, that's one of the, I'm not aware of other combination therapies doing that. The other combination therapies are effective because they give more of the same cell. So you get many more cells, and there's PD-1 blockade at the target site, so you get viral control. But this is the first clear demonstration that the differentiation trajectory of the stem-like cells can be modified to give you much better effector cells. I think that's a major finding in the field, that we can actually change the exhaustion program. Because until now, the feeling of the community was that that's kind of fate locked. You can get more effector-like cells, but still it is the exhaustion program. And that's true. That's what happens with the PD-1 monotherapy. But as Masao shows very nicely in this paper, but when you do the combination therapy, it's really a very different ballgame with getting much better. So I think to me, that's a major finding. So far, the response we have gotten to the paper by people emailing or calling, I think they're all very impressed by that, by that finding of Masao's. The other thing that you pointed out very nicely is showing the importance of engaging CD25 to really harness the IL-2 effect. And as you are well aware, the field currently is kind of focused on making IL-2 molecules, or using IL-2 molecules that bypass CD25 binding. There are some good reasons for this. The issue that you brought up about activating Tregs, also the issue you brought up about potential side effects. So there are safety concerns. But a couple of things that we also show in this paper is that when we use the wild-type IL-2, you do expand effector cells. I mean, you do expand Tregs, but the expansion of the specific cells is significantly greater. So the ratio of Treg to effector actually improves. That is many more effectors to Treg. So Treg expansion is about twofold to threefold in these experiments. Effector cell expansion is like 50-fold.
Yeah.
So now we don't know whether the same thing will happen in human cancer. It may be slightly different there, the ratio of the two, but at least in this model, the effect is much greater on this effector cells that will actually control the infection of control the tumor. I think that's really quite a promising and interesting result. And then the last point is that you pointed out is that if you use the mutated IL-2B, you pretty much lose the synergy between the cytokine and PD-1 blockade. And this data that Masao generated beautifully ended up in the supplementary figures. But the mutated cytokine is biologically active in vivo. So it will expand a lot of CD8 T cells. So if you look at total CD8 T cell expansion after giving either the wild type IL-2 or the mutated IL-2 that doesn't bind CD25, the overall CD8 T cell expansion is very similar. But the IL-2 mutated one is just expanding all the other total CD8 T cells. The selectivity is totally gone. Whereas these cells, as you pointed out, because they express the very high affinity trimeric receptor, they just capture all the IL-2 and get the most optimal signal. So there you're getting the sink effect. It gets all diluted out and it's expanding every cell that has the beta chain and the gamma chain, which is every T cell in the body. As Masao pointed out, the frequency of that specific cells is very small. With the wild type IL-2 expressing CD25, because these cells very quickly express high levels of trimeric receptor, also shown in a supplementary figure in the paper, they just capture it. And then they expand and capture most of those. So the effect on the other cells is very minimal, is the antigen-specific cells with the high levels of CD25 and the trimeric receptor basically capturing all the IL-2. So I think there are, as you pointed out, there are some lessons for people doing the clinical trials to think about either generating something which is still tolerable but binds CD25, or targeting it to the cells of interest. And in this companion paper that Masao also played a major role in, he's the co-first author of the paper from Roche, which is the companion paper. The Roche strategy was to have an anti-PD-1 antibody to which they have IL-2, the mutated IL-2. So in that case, we do get good responses. And the reason for that is because now the IL-2 is only targeting the PD-1 positive cells.
Yeah, yeah.
So I think that the field has to change into either the IL-2 field in terms of clinical trials has to change either in terms of a safe way of engaging CD25 or using a targeting strategy so it gets to the right cell. Otherwise, these things are unlikely to work. And you're very familiar with what happened to Nectar. You know, it was very much, there was a lot of promise and a lot of hope that the speculated IL-2 made by Nectar would work. But of course, it did not work. Not surprising to us based on our results. And essentially, the company is gone now. The company doesn't exist. In spite of the huge investment of BMS. I think BMS invested over $1 billion on the pegylated interferon. Pegylation certainly increased the half-life, which was beneficial. But again, that cytokine was, the pegylation was done in such a way that it did not.
Yeah, the pegylation made it so that it didn't bind with high affinity to the trimeric receptor.
I think that failed. We're not surprised at all. You know, so I think that the field has to kind of rethink a little bit that making molecules that don't bind alpha chain at all and are not being targeted. We have so many T cells, they all suck it up. You know,
yeah, and that all those other T cells that get activated through the beta gamma chain, the beta gamma chain on those cells are really IL-15 receptors that are expressed on memory T cells. So they get all these memory T cells, but they don't do anything for the virus.
Yeah, also the naive cells, all the naive cells. So you're just expanding everything. And we showed that, Masao shows that in the supplementary figure that it is, IL-2B is doing what it's doing, but it has lost the specificity for the key molecule, which is the alpha chain and the trimeric receptor. As you showed nicely, the trimeric receptor is the high affinity.
You know, I think because I think you probably know that during the 90s, I tried to develop what I call low-dose IL-2 for the treatment of HIV infection. And I worked out all kinds of experiments with the pharmacokinetics and the pharmacodynamics, and related that to the affinity of the IL-2 receptor. And basically, the field ignored that. And that was in HIV. And the cancer people, because they started out with very high dose, they did almost 100-fold higher doses of IL-2 given in an intravenous push injection. It just
so we're hoping that
I think
the field will pay some attention to it and try and either use lower dose of IL-2 wild type or modify IL-2 in such a way that it's still binding to the alpha chain, but maybe reduce toxicity in some way, or targeting it to the cells of interest. So we are hoping that at least it will have some impact on the clinic in terms of as we move forward. And I agree with you that I think that IL-2 is probably the best partner in terms of providing positive signals. It is, as you and others have shown, it is the key effector cytokine. For CD8 T cells, it is the key effector cytokine.
Yeah, music to my ears, Rafi. Have you had any feedback yet from the community and
it's been very positive, actually, very positive, even from people who are doing very elegant work in modifying cytokines. So so the cytokine biologists, molecular biologists are also paying attention, and we've gotten some very positive feedback from these very nice scientists.
That's good. Yeah, that's great.
And Masao has been engaged also in in discussions with you. So it's been very satisfying. So Masao's amazing commitment and hard work.
Really, and one of the things I was going to say when Masao was talking was, I couldn't believe how many little questions all the reviewers of the paper had, and they wanted you to do this, and they wanted you to do that.
I know.
I went on and on and on.
And you saw the revised paper that we submitted. It had 30 extended data figures, 30. And the paper was about 3 times, would have been like 20 pages of 25 pages of nature. So that version was accepted. And then it took Masao and I almost 2 months to cut the thing down. It was not that easy to cut it to the size they wanted.
Yeah, well, I can imagine. Yeah.
Yeah, but anyway, it's even though it's taken longer than it should have, a lot of the blame goes to me initially because being slow in writing it up. But the story was very elegant and the data just fantastic. So we wanted to be sure the right message comes across. And I think we've been successful in that based on the response we have had to the paper.
Well, going forward, where do you go from here? It seems like you've wrapped this up with a bow and you can send it off for a Christmas present.
Yeah, for this part, yes. But again, Masao has many things he's looking at. Maybe Masao can tell you about some of the next questions. Masao, please go ahead. We have just maybe a few minutes left, so you can comment on that.
Yeah, next question. I mean, so one big question is we can see the same phenotypic or transcriptional or epigenetic change in human cancer, right? Basically, PD-1 plus IL-2 combinations exert some similar.
Yeah, I was going to ask that. Yeah.
That is a kind of translational question. Then one other scientific and basic scientific question is how long, I mean, effector cells generated by IL-2 will persist after cessation of the treatment. That's another question. And then a lot of detailed questions are there. So maybe we can figure out what
I
is.
think these are the two. Masao highlighted it very nicely. We are, Kendall, you'll be pleased to know we are planning a small clinical trial in lung cancer patients who will get PD-1 blockade and the low dose IL-2 that you have recommended and used. And so this will give us an opportunity to see whether the low dose IL-2 plus PD-1 blockade.
Yeah, that would be great.
This is the wild type IL-2 cytokine, okay, and see whether we see something similar. And the other one that Masao mentioned is since these new effector-like cells that are generated have a very different transcription and epigenetic program, more resembling what we see during acute infection. The big question is, what is the durability of this?
Yeah.
What's the durability? Can these cells live for a longer period and be effective? So we'll address the durability issue as a LCMD project, but then we really want to see what happens in humans with this low dose IL-2 plus NTP.
If you haven't, you haven't started those trials yet, is that correct?
No, we haven't. No, we are planning to start them soon. We are doing this with Dr. Suresh Ramalingam, who's a lung cancer oncologist at our cancer center, the Winship Cancer Center. He's also the director of the cancer.
Where?
No, he's also the director of the Emory.
At Emory. Yeah. Well, if you want, when you get your protocols, fleshed out, if you want me to look at the therapy, I'd be happy to.
Absolutely, thank you, Kendall. We would appreciate your comments.
Because I could save you some time. The extrapolation from the mouse doses to the human is essentially you can't really extrapolate because
I know that, right. Yeah, we'll run it by you. We'll run it by you.
Yeah. Okay. Well,
great.
Yeah. Good luck.
Yeah. Thank you for this opportunity. And we'll of course stay engaged.
Well, thank you so much, both of you. I hope I keep on trucking, you guys. You're doing a great job.
Yes.
Yeah. Thank you, Kendall. Yeah.
Thank you. Okay. All right. Thanks, Vincent.
Okay. Bye-bye.
Bye-bye.
If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.